Hello students, I'm Shifan, and in this video we'll be going through the 2023 G.C.E level biology MCQ paper. Now on our last video series we went through the 2024 biology MCQ paper and a lot of students responded to that. Thank you so much. And in this video we'll be analyzing the 2023 paper. Now if you haven't watched the 2024 MCQ analysis, the link for that playlist will be given below. If you want to, you watch that too. Now let's go through the 2023 paper. Okay. So the first question on the 2023 paper was about organelles present inside the cell, specifically lysosomes and peroxisomes. So the question was a feature common to lysosomes and peroxisomes is that they. Now we know lysosomes and peroxisomes are present inside the cell. So lysosomes are usually involved in autolysis, then digesting worn-out organelles and exocytosis. And when you take peroxisome, they are usually involved in detoxification. But something common in both of these organelles is that they are single membrane bound organelles. So as the first statement says, this is the correct answer. Okay. Now let's go through all the other answers too. Transporting residual materials by exocytosis. Now, as I told earlier, lysosomes are involved in digesting worn-out organelles. So they kill the old organelles that are present inside the cell. So these old organelles, when they are digested, the waste materials should be secreted out of the cell. So lysosomes are vesicles. So what they do is they engulf these unwanted materials and they go and fuse with the cell membrane. They fuse with the cell membrane and open up the substances to the extracellular matrix. So transporting residual materials is a function of the lysosomes. So I'll write that down. Okay. Contain oxidizing. So third statement, contain oxidizing enzymes that catalyze breakdown of nucleic acid. Now we know lysosomes are involved in autolysis, which means they cause the cell death. We know after a certain time the cell will get old. When the cell gets old, the cell has to die, right? For this reason, lysosomes are present. Lysosome causes cell death. Now, during cell death, everything inside the cell is broken down. So are the nucleus and the DNA that is present in the nucleus. So lysosomes contain enzymes to break down the nucleus. So the third statement is a function of the lysosomes. And another important thing is that lysosomes doesn't have oxidizing enzymes. They have hydrolytic enzymes. So oxidizing enzymes, they are present in peroxisomes, not lysosomes. So this is a function of the lysosome, but they use hydrolytic enzyme. Then the fourth statement are important in photorespiration. Now, when it comes to photorespiration, the organelle that is present inside the cell that is involved in it is peroxisomes. So along with mitochondria, chloroplast, peroxisome is the other organelle that is involved in photorespiration. So there are three organelles. One is mitochondria, then we have chloroplast, and we have peroxisome. Now, again, fifth answer, digesting worn-out organelles. We know digesting worn-out organelles is a function of the lysosomes. So the correct answer is the first answer. Now let's go through the second question. Okay. Two characteristics that can be seen only in living organisms are. Okay. Now, so the first statement they have given adaptation and growth. So we know adaptation is the ability of an organism. So it is the unique features that an organism contains that will allow them to survive and reproduce. So adaptation is only present in living organisms. Non-living organisms, they don't adapt to the environment, the conditions. So adaptation is correct. But growth. So let's write the definition of adaptation. Okay. What about growth? Now, the word growth has two definitions. It has a general definition and it has a biological definition. That's why we call the word growth ambiguous, right? Because they have two definitions. Now, the biological definition is increase in dry mass. Yes, it occurs in living organisms. But the most used meaning of the growth is the general meaning, which means to get bigger, right? So if you take the meaning to get bigger, even non-living organisms gets bigger, right? So if you take, for example, the best example is crystals. If you take crystal, they get bigger by the chemical reactions that occur inside them. They get bigger, but they are non-living organisms. And if you take another easy example, let's tell a snowball, right? A snowball, when rolling down the snow, it starts to get bigger, right? It starts to grow. So that is also an example of growth. For that reason, we can't take this answer, because the word growth has two different meanings and it can also be seen in non-living organisms. Now let's go to the second statement, movement and irritability. Now again, movement. Even non-living things can move, right? If you take the wind, wind moves, the water, water moves, and if you also take machines, machines also can move. So it is not something only related to living organism. Then we have irritability. So what is irritability? So irritability is the ability of an organism to respond to a stimuli. So let's write that down. Ability of an organism to respond to a stimuli. So only living things can respond to the stimuli that is present in the external environment, right? So even though irritability is correct, movement is wrong. For that reason, we eliminate the second answer. Then we have change with time and development. Now development. So development is the increase in the irreversible mass that occurs through the lifetime of an organism. Now development, it is a characteristic of a living organism, which is correct. But what about change with time? Right? If you take mountains, mountains change with time, right? If you take rock, a rock will change with time. So your fossil fuels change with time. So these are all non-living things. For that reason, we eliminate this answer. Then fourth one, we have metabolism and heredity. Now metabolism is a chemical reactions that is taking place inside the cell, right? We can store substances, we can release substances. So metabolism is only present in living organisms, not non-living organisms. Then we have heredity. So heredity is the ability of an organism to pass down gene from the parent to an offspring, and this is also another characteristic that can only be seen in living organism. So the fourth one is the correct answer, and I'll highlight it. Okay, finally we have synthesis and decomposition. Synthesis is the making up of substances, right? So synthesis can also be seen in non-living things, and decomposition also can be seen in non-living things. So decomposition is the breaking down of substances. So most of the chemical reactions, they break down substances, and it can also be seen in non-living organisms, sorry, non-living things, which is wrong. So the correct answer is the fourth one. Now let's go through the third question. Which of the following statements is correct regarding transmission electron microscope? Now, when it comes to microscope, we have two types. We have the light microscope and we have the electron microscope. In an electron microscope, we have again two types. We have the transmission and the scanning electron microscope. So this question is regarding transmission electron microscope. So they are saying specimens are magnified 5 into 10 to the power 6 times. But in transmission electron microscope, the specimen is magnified 5 into 10 to the power 5 times, not 6 times. But theoretically, they can have a magnification of 1 into 10 to the power 8 times. Also remember that. 1 into 10 to the power 8 times. So this is the theoretical magnification. Now let's go to the second statement. Less electrons may get displayed in cellular structures with dense staining. Now we know in transmission electron microscope, we use heavy metals to stain the organelles. So we usually stain the mitochondria, the chloroplast, or the ribosome. So these are all organelles, and we stain them. So when we stain them, the electrons can't pass through them, and they start to scatter. Only the unstained part only in the unstained part the electrons will pass through. So on the final image, on the display we'll get bright, bright areas and dark areas. So the dark areas means it is the organelles because the electrons didn't pass through those areas. So this statement is saying less electrons may get displayed in cellular structures with dense staining. Now, in the normal transmission electron microscope, this statement is correct. Okay, so in a bright field electron microscope, as I told, most of the electrons pass through and some electrons get scattered. Okay, so in a bright field electron microscope, this is correct. So this is out of syllabus, but I'm explaining it so that you guys will get an idea. But here this statement, they are talking about the dark field electron microscope. In a dark field electron microscope, what really happens is, as I said, when we stain it with heavy metals, the electrons get scattered, right? So the scattered electrons are used to form the image. So the scattered electrons have scattered image has the more electrons. So this statement is wrong according to dark field transmission electron microscope. So I know I'm confusing you guys, but always remember, more electrons may get displayed in cellular structures with dense staining in dark field transmission electron microscope. So this statement is wrong. And third statement, living specimens cannot be observed. Yes, this is correct because the living specimen should be killed to be viewed in a transmission electron microscope. Why? Because to see a cell in a transmission electron microscope, it has to be cut into very thin pieces. So in this process, the cell dies. And again, all the what is dehydrated in the cell to be viewed in the transmission electron microscope. So when the cell is dehydrated, again the cell dies. So this is the correct statement. So I'll highlight it. Live specimens can be observed. The fourth answer is three-dimensional appearance of the specimen can be observed. So in transmission electron microscope, only two-dimensional images can be seen. But on scanning electron microscope, which is another type of electron microscope, 3D images can be viewed. So this statement is wrong for transmission electron microscope. And fifth answer, specimens scatter many electrons while others are observed. Okay, so in a transmission electron microscope, only some electrons are scattered. So these electrons go and knock over the stained areas, which are usually the organelles. So some electrons scatter and many are observed. So this statement is wrong. But this statement is actually correct for a scanning electron microscope. So in a scanning electron microscope, specimens scatter many electrons while others are observed. So the correct answer is the third one. Now let's go to the fourth question. Okay, select the response which correctly indicates the event and phase in the eukaryotic cell cycle. Okay, the question is regarding the cell cycle. We know the cell cycle has two phases. We have the interphase and the mitotic phase, right? So the G1 phase, S phase, and G2 phase occurs in the interphase. Now they are saying DNA replication occurs in the G0 phase. Now G0 phase is the non-mitotic phase. So in the non-mitotic phase, cell division doesn't occur. So the cell doesn't undergo G1 phase, G2 phase, any other phases cell doesn't undergo because it is in the non-mitotic phase. So this is called the non-mitotic phase. Cell doesn't undergo cell division here. So this first statement is wrong. They say DNA replication occurs, so DNA replication doesn't take place. And second statement, synthesis of proteins. Now, G1 phase is the first phase of the cell cycle, and second statement is correct because a lot of proteins are synthesis for in the G1 phase. So what are these proteins? These are usually proteins that are required for DNA replication, right? So we know for mitotic division to occur, first the DNA should be replicated in the interphase. So all the proteins that are required for DNA replication, such as polymerase, helicase, primase, so these are all synthesized in the G1 phase. So I'll write it proteins. So second statement is correct, and I highlight it. Okay. Sorry. Okay, now let's go through the other statement. Chromatin formation. Now, as I said, DNA replication occurs in the interphase. So when the DNA replication occurs, the DNA should be arranged, right? So that's where the chromatin formation comes in. And after G1 phase, we have the S phase. So in the S phase, chromatin formation occurs not in the G2 phase. So chromatin formation occurs in the S phase. So third statement is wrong. And fourth one, production of cellular organelles. We know this occurs because we have to form two cells, right? During mitotic division, one cell is divided into two. So the other cell also should have all the organelles such as the mitochondria, the ribosome, the Golgi apparatus. So all this production of organelles occur, but this occur initially in the G1 phase, not in the S phase. So again in the G1 phase, another thing occurs, that is the production of cellular organelles. Finally, we had duplication of the centrosome. Okay, centrosome formation occurs in the S phase, not in the M phase. In the M phase, what occurs is the M phase is the prophase, metaphase, anaphase, telophase. So those stages are in the M phase, and centrosome occurs before all these stages. So it occurs in the S phase. Sorry, G2 phase. Okay, the correct statement is the second one. Now let's go through the fifth question. In allostatic regulation of enzymes, so the question is regarding allostatic regulation. Now let's go through a quick summary of what allostatic regulation is. So, allostatic regulation is the activation and inhibition of the enzyme by using certain allostatic molecules such as the activator and the inhibitor, which can activate or inhibit the enzyme. Now, here I have given allostatic inhibition. So during allostatic inhibition, what happens? Normally, the enzyme is active, it can bind to the substrate. But when an inhibitor molecule binds to the allostatic site, so this site we call it the allostatic site. When the inhibitor molecule binds to the allostatic site, the enzyme changes its 3D shape. So when the enzyme changes its shape, it becomes inactive. It changes the shape of the active site. So the substrate can't bind to the active site. So no reaction takes place. Then again, the opposite occurs in allostatic activation. Here the enzyme is inactive. But when activator binds to the enzyme, it activates the enzyme. It changes the shape of the enzyme. So when the activator binds, active site is working and substrate can bind to the active site and the product is formed. So this is the summary. So now let's go through the answers. Regulatory molecules bind reversibly to the active site of the enzyme. So we know this inhibitor and activator is the regulatory molecule, and they bind to the allostatic site, not the active site. So we can eliminate this answer. Second statement says regulatory molecules bind to the enzyme via non-covalent interaction. We know this activator and inhibitor, they don't bind forever, they bind reversibly. So to bind reversibly, they should have non-covalent interactions. So the second statement is actually correct. So I'll highlight it. Now let's go through the third, fourth, and fifth statement anyways. So the third statement says an activator molecule that binds to a particular subunit will affect the active site of that subunit only. Now, in reality, if we take an enzyme, that enzyme has more than one active site. They might have 3 active sites, 4 active sites, but here in this image, we only have 1 active site. So when an activator binds to an enzyme, all the active sites are activated. Again, the opposite, if an inhibitor binds to the active site, all the active sites are inhibited. So this statement is also wrong. Fourth statement, inhibitory molecules affect the function of the enzyme, not the shape of the enzyme. We can see when the inhibitor binds to the enzyme, the shape of the enzyme also changes, right? So shape of the enzyme changes, shape of the active site also changes. So that statement is also wrong. And the fifth statement, ATP functions as an allosteric activator. Now, we know ADP is used to form ATP, right? So between the reaction, we have an enzyme that catalyzes that reaction. Now ADP binds to the enzyme and forms ATP. So when there's too much ATP acts as an allosteric inhibitor, not as an activator. So when too much ATP is produced, ATP go and bind to the enzyme as an inhibitor and inactivate the enzyme. So no more ADP is converted to ATP. So this statement is wrong because ATP functions as a allostatic inhibitor, not activator. Now let's go to the sixth question. So it's about ethyl alcohol fermentation. We know ethyl alcohol fermentation is anaerobic respiration, right? So first statement says one molecule of glucose produces one molecule of pyruvate and two molecules of NADH. Now we know glucose is a 6-carbon molecule. So when glucose is broken down, it breaks down to two pyruvate molecules. So we know pyruvate is 3-carbon. So glucose breaks down to two pyruvates and two molecules of NADH. So the first statement is wrong. Then second statement, pyruvate is reduced directly to ethanol using NADH. We know in ethyl alcohol fermentation, pyruvate is first reduced to acetaldehyde. Right? Pyruvate is 3-carbon, and by releasing carbon dioxide molecule, they convert to acetaldehyde, which is a 2-carbon molecule. So this statement is also wrong. So the process, let's go through the process. So pyruvate is converted to acetaldehyde and acetaldehyde is converted to ethanol releasing 2-NADH. So this statement is wrong. Third statement, one molecule of carbon dioxide is produced from one molecule of glucose. Now, one molecule of pyruvate is converted to one molecule of acetaldehyde. So during this reaction, one carbon dioxide is produced. But in a glucose molecule, we have two pyruvate molecules. So in reality, two carbon dioxide molecules are released, not one molecule of carbon dioxide. So again, third statement is also wrong. Fourth statement, phenyl hydrogen acceptor is an inorganic compound. Now, pyruvate is converted to acetaldehyde, acetaldehyde is converted to ethanol, right? So the phenyl hydrogen acceptor is the acetaldehyde, right? Because during the reaction, acetaldehyde to ethanol, NADH is converted to NAD+. So hydrogen acceptor is the acetaldehyde. And acetaldehyde is not an inorganic compound. Acetaldehyde is an organic compound. So fourth statement is also wrong. And the final statement, two molecules of ATP are produced from one molecule of glucose. Okay, when glucose is broken to pyruvate, 4 ATP are produced, but 2 ATP are used up for the reaction. So 2 ATP molecules are produced from one molecule of glucose. So this is the correct answer. Now let's go through the seventh question. Which of the following statements regarding glycolysis of one molecule of glucose is correct? Again, this is about glycolysis. So we know glycolysis is the first process that occurs in aerobic respiration. So there's a net yield of 4 ATP molecules. Yes, during glycolysis, glucose is broken down to 2 pyruvate molecules. And during this process, this is wrong actually, because during the process, 2 ATP molecules are produced. 4 ATP molecules are produced and 2 ATP molecules are used up for the reaction. So the net yield is 2 ATP molecules, not 4 ATP molecules. So this statement is wrong. And second statement, 2 hydrogen ions are released. So in glycolysis, 4 hydrogen ions are released, not 2 hydrogen ions. Again, that statement is also wrong. And third one, it partially depends on molecular oxygen. We know glycolysis is independent of oxygen. So it takes place whether oxygen is present or not present, because it doesn't need oxygen. So that statement is also wrong. And fourth statement, 2 NADH molecules are formed. So yes, during glycolysis, glucose is converted to pyruvate. During that reaction, 2 NAD plus is converted to 2 NADH. So this statement is correct. Now let's go through the final statement. Part of glycolysis takes place in the outer membrane of mitochondria. Now, glycolysis occurs in the cytosol. Why? Because all the enzymes that is required for glycolysis to take place is present in the cytosol. But after glycolysis, we have the other two processes, which is the Krebs cycle and the electron transport chain. Both of them which occurs in the matrix of mitochondria. But glycolysis, it occurs in the cytosol. So the correct answer is the fourth one. Okay, now let's go to the eighth question. Some events that took place during the evolution of organisms are as follows. So they have given saturation of water bodies with oxygen, oxidation of iron, increase in photosynthetic bacterial population. So all the first three events to occur, first we need oxygen, right? So oxygen, where did oxygen initially come from? It comes from cyanobacteria. So the first statement is origin of cyanobacteria. Why? Because cyanobacteria were the first organism to carry out oxygenic photosynthesis. Because there were organisms that carried out photosynthesis, but oxygen was not produced. But cyanobacteria was the first organism that produced oxygen as a result of photosynthesis. Now, what happens? So when cyanobacteria released oxygen, so this occurred in the water. So during this period, there was no land animals or anything on the land. So these all occurred in the water. So cyanobacteria released oxygen in the water. Then what happens? There were a lot of iron inside the water. So oxidation of iron took place. So first statement is the first statement is the D1, and the second statement is the B1. So what happens? Oxidation of iron took place and iron reduced from Fe2 plus to Fe3 plus. Now, so this process carried out for years and years, but after a certain period, there was no more iron in the water. Now what happened? There is no iron in the water, so the oxygen is not used up. So the oxygen started to get saturated in the water. So the third one is the A, saturation of water in the water bodies with oxygen. Finally, cyanobacteria start to reproduce and photosynthetic bacterial population start to increase. So that is the fourth one. So the correct order is D, B, A and C. So D, B, A and C, which is the fifth answer. So fifth answer is the correct one. Okay, now let's go to the ninth question. Okay. Which of the following pairs of organisms have the highest number of common characteristics? So we know the biological hierarchy, right? So from domain, kingdom, phylum, class, family, genus, species, so it moves down. So as we move down, number of highest common characteristics increases, right? So as we move down, so number of organisms having more common characteristics increases. So for example, two organisms in the same class has more common features than two organisms in the same phylum. So again, two organisms in the same phylum has more common characteristics than two organisms in the same kingdom. So that's how it works. So first statement, we have bat and a crow. Okay, now let's answer this. Okay, what happens? Okay, now bat and crow both belong to phylum Chordata, right? But bat belongs to phylum Amphibia and not crow. Okay, crows belong to phylum class A. So even though they are the same phylum, they belong to different classes. So they don't have the highest number of common characteristics. Then second, we have lizard and turtle. Now both lizard and turtle are from phylum Chordata and they are from the same class too. So the class is Reptilia. So they have the highest number of common characteristics for now. So anyways, let's go through the other answers. So they both are from the same class, highest number of common characteristics. Then we have Ichthyophis and Taenia. Again, Ichthyophis, they belong to phylum Chordata in but class Amphibia. So phylum Chordata. What about Taenia? So Taenia belongs to phylum Platyhelminths. So these both are from two different phylums, so they have the least number of common characteristics. Okay, now we have Ulva and Pogonatum. Now Ulva is from another phylum. Ulva belongs to phylum Protista. Kingdom Protista. So they have Kingdom Protista. Okay. And Pogonatum belongs to Kingdom Plantae. Right? So they have the least common characteristics, right? Because as we move up, the common characteristics decreases. So kingdom plant and kingdom protista are two different things. So the common characteristics decreases. Then we have Pinus and Cycas. So Pinus and Cycas, they belong to kingdom Plantae, but Pinus belong to phylum Coniferophyta, and Cycas belong to phylum Cycadophyta. So lizard and turtle is the correct answer because they belong to the same class. So organisms that are present in the same class have the highest number of common characteristics. So the correct answer is the second one. Now let's go through the 10th question. Which of the following are unique characteristics of some phyla of kingdom Animalia? Okay, they have given 4 features: internal fertilization, parapodia, radula, and nephridia. Now, is internal fertilization a unique characteristic? No, right? Because a lot of phyla has internal fertilization. So if you take nematoda has internal fertilization, then erythropoda has internal fertilization, and also chordata also has internal fertilization. But what about parapodia? So parapodia is only present in phylum Annelids. So that is a unique feature. So parapodia is correct. And radula. So radula are like tooth tongues, they say, right? So radula is present only in annelids. Sorry, in mollusks, which is correct. And finally, nephridia. So is nephridia a unique feature? It seems like unique feature, right? But is it? So nephridia is present in annelids, right? So it can be a unique feature, but the thing is, nephridia is also present in platyhelminths. So platyhelminths also carry a form of nephridia, which we call it the protonephridia. So again, it is not unique. They carry something called the protonephridia. So the correct answer is parapodia and radula. So which is B and C, which is the third answer. Okay, now let's go through the 11th question. Select the correct statement regarding vascular tissues of plants. Now, if we take vascular tissues, so first statement saying xylem tissues of pterophytes contain tracheids. So pterophytes, they are seedless vascular plants. So let me write it down. Now, in seedless vascular plants, they have xylem vessels. Okay, xylem vessels are present. But the thing is, they don't have one specific element of the xylem vessels, which is the vessel elements. But they do have the tracheids and the parenchyma cells and the fibers, so first statement is correct. So always remember, pterophytes, which are seedless vascular plants, they don't have the vessel elements, but they have the tracheids. Second statement, xylem vessel elements are long and tapered cell. Now, we know xylem has 2 tubes, they have the vessel elements and we have the tracheids. Now, tracheids are the ones that have the long and tapered cells, which looks like this. So we know, so this is how a tracheid looks like. So I'm not good at drawing anyways. So these are the tapered ends of the tracheid. So second statement is wrong because tracheids are the one with the tapered cells. Okay, now let's go to the third statement. Tracheids provide support to the stems of bryophytes. So bryophytes are non-vascular plants. So when they are non-vascular plants, they don't have a vascular system. When they don't have vascular system, they don't have xylem or phloem vessels. So they don't have the tracheids. So again, that is wrong. Then fourth statement, companion cells are found in Cycadophyta. So another thing to learn here. Now, when it comes to, so Cycadophyta is a gymnosperm, right? So another thing, in gymnosperms, they don't have the vessel elements and the companion cells. So these are 2 things that are not present in gymnosperms and also in seedless vascular plants. So even in seedless vascular plants, they don't have companion cells. So this is something you should remember. Okay, so this statement is wrong because Icodophyta is as I said, it's a gymnosperm and they don't have companion cells. Finally, pits are present between sieve tube elements. So sieve tube elements, they belong to phloem vessels, and pits are not present between the sieve tube elements. Pits are usually present in the vessel elements and the tracheids, which are found in the xylem vessel. So first one is the correct answer. Okay, now let's go to the 12th statement. Some structures of plants and their function are shown below. So this is a very tricky question, because we know both lenticels and stomata carry out gas exchange and transpiration, right? Even though lenticels carry out a very little amount of transpiration and gas exchange, which is 5%, and stomata carries out 95%, they carry out transpiration and gas exchange. But they don't carry out guttation, right? So guttation is only done through the hydrothotes. So that's something we can be sure of, right? So let's see, we have two answers that goes with C and R. So that is the third answer and the fifth answer. So it's got to be either one of these. So in third answer, A goes with P, lenticels transpiration, which is correct. B, Q, stomata, again with gas exchange, yes, which is correct. Then C goes with R, hydrothotes, guttation. So this is the correct answer. Anyways, let's go to the fifth one. So in fifth question, A goes with R. Now, lenticels carry out transpiration and gas exchange, which is correct, but they don't carry out guttation. Because of that, this statement is wrong. So the correct answer is the third one. Okay, now let's go through the 13th question. Transport of water molecules due to physical adsorption by hydrophilic material is called. Now, we know whenever the word adsorption comes, the method of transport is imbibition, right? Because during imbibition, what happens? Hydrophilic material go and adsorb to water. So this water goes and attached to the surface of the hydrophilic material. So these hydrophilic material are usually cellulose that are present in seeds. So which this will cause a substance to swell up. So the best example is seed germination, right? So during seed germination, so there is a seed which contains hydrophilic material such as cellulose inside them. So what happens? The water molecules go and absorb to their surface. So the water molecules go and absorb to the surface and cause the seed to swell up. So when the seed swell up, it causes the seed to break open and cause germination. So that's what imbibition is. So it is the correct answer. Anyways, let's go through what all these other terms are. We have osmosis, which is also type of diffusion, but occurs between water molecules from a high water potential to low water potential. And again, we have facilitated diffusion. Again, it is also another type of diffusion, but of course through transport membranes, which are present between cell membranes. Again, fifth one and fourth one, sorry, I have changed it. Fourth one and fifth one, bulk flow and mass flow. So bulk flow and mass flow also refers to the same thing, where bulk movement of substances with water, which occurs due to pressure gradient. So the correct answer is the first one. Okay, now let's go through the 14th question. Some steps in the process of opening and closing of stomata are given below. So this is regarding the opening and closing of stomata using the guard cells. So let's see the right order. So in all the statements they have given us, A occurs first, which is correct, flow of water into the guard cells. So let us mark it. So what happens is, so when the stomata is closed, potassium ion starts to move inside the stomata, start to accumulate inside the guard cells. So when potassium ions accumulate in the guard cell, the water potential in the guard cell decreases and water starts to move in. So which is correct, which is the first one. What happens after the water starts to move in? Water starts to move in and the turgor in the guard cell start to increase. The pressure inside the guard cell start to increase and causes the expansion of the guard cell. So which is the third one. So during the expansion of the guard cell, we know guard cell expand non-uniformly, which means the outer wall expand perfectly, but the inner wall they don't expand because they have a thinner wall, very thicker wall, sorry. So this thick wall starts to bend. So this is the second one, bending of the inner walls of guard cells. Then what happens? The stomata opens. So when the stomata is closing, again the potassium ions leaves the cell. So with the potassium ion, water leaves the cell. So when water leaves the cell, the turgor pressure inside the guard cells decreases. So here pore opens, then water leaves the guard, turgor in the guard cells decreases. Then finally what happens, when the turgor decreases, the water leaves and everything, the stomata closes, the guard cells closes. So that is the final answer. So this is the fifth one, and this is the sixth one. Sorry, yes. So the correct order is 1 A C B D E F, A C B D F. So it is the second answer. So I'll highlight it. Anyways, let's go through the 15th question. Okay. A macronutrition and a micronutrition which causes chlorosis in plants due to deficiency are respectively. Okay, we know macronutrients are substances that a plant need in very large amounts, and micronutrients are substances, nutrients that a plants need in very little amount. And chlorosis is the yellowing of plants due to deficiency of chlorophyll. Now, first let's take a look at all the macronutrients and then the micronutrients. Then let's see what causes chlorosis. So the macronutrients. Okay. So first three obvious macronutrients are NPK, which is the nitrogen, phosphorus, and potassium. Then we have the organic macronutrients. We have carbon, oxygen, and hydrogen. Then we have magnesium, sulfur, and calcium, right? So and calcium. So the micronutrients that are present inside the cell are chlorine, iron, manganese, boron. So let me write down chlorine, iron, boron. Then we have zinc, copper, nickel, molybdenum. And molybdenum. Now, these are all the macronutrients and the micronutrients that are present inside the that is required for the plant. So, which causes chlorosis? So, chlorosis is caused by nitrogen, magnesium, sulfur, iron. So, let me write it down. Nitrogen, magnesium, sulfur, iron, right? Then what else? Then we have copper, not copper, sorry. Then we have molybdenum, nickel, and magnesium. Magnesium, okay. Now, first statement we have magnesium and molybdenum. Now, magnesium is a macronutrition, which is correct, and molybdenum is a micronutrition. And both magnesium and molybdenum are present that causes chlorosis. So, first one is the correct answer. Anyways, let's go through all the other answers. We have iron and nickel. Yes, iron causes chlorosis and nickel causes chlorosis, which is correct. But what is wrong here? Iron is not a macronutrition. Iron is a micronutrition. And nickel is also another micronutrition, so that is wrong. Then we have phosphor and molybdenum. Yes, phosphor is a macronutrition, but it doesn't cause chlorosis. Then we have molybdenum. Molybdenum causes chlorosis, but molybdenum causes chlorosis, but it is a macronutrition. I have wrote it there. Let me check. So molybdenum is a micronutrition. So let me write it down. Is a micronutrition. So phosphorus, yes, phosphorus is a macronutrient, but again no chlorosis. And molybdenum is a micronutrition, but this statement is wrong because of phosphor. Then we have nitrogen and sulfur. Again, nitrogen and sulfur both are macronutrition, so we can eliminate that answer. Then we have copper and boron. Okay, copper is not involved in chlorosis, and boron is also not involved in chlorosis. So the correct answer is the first one. Two plant hormones that promote root formation. Okay, first we have auxin. Yes, auxin is one of the main hormones that causes plant formation, which is correct. But gibberellins are usually involved in stem elongation and seed germination. For that reason, we can eliminate the first answer. Then we have cytokinins and abscisic acid. So cytokinins, again, cytokinins are involved in stem elongation, not root formation, but they do have a role in cell division in the root, but they don't promote root formation. Then we have abscisic acid. So abscisic acid, they are usually involved in inhibiting growth, so they don't form the roots. So again, first statement is wrong, second statement is wrong. Then we have ethylene and auxin. Okay, ethylene is another hormone that causes the root formation and root hair formation, which is correct. And auxin is also it's another hormone that causes root formation. So the third one is the correct answer. Fourth one, ethylene and gibberellin. So ethylene, yes, it forms root formation, but gibberellins, their function is stem elongation. So again, we can eliminate that answer. And fifth one also we can eliminate, because cytokinins again involved in stem elongation, and gibberellins also stem elongation, not root formation. The correct answer is the third one. Now let's go through the 17th question. Which of the following statements regarding epithelia is correct? We know humans have four kinds of tissues, right? We have the epithelial tissue, connective tissue, muscle tissue, and the nervous tissue. So in the epithelial tissue, we have two types of epithelial tissue. We have simple epithelial tissue and compound epithelial tissue. And in simple epithelial tissue, we have simple squamous, simple cuboidal, simple columnar, and pseudo-stratified columnar. And in compound epithelial tissue, we have stratified squamous, and we also have another one called the transitional. Okay, now let's go through the first statement. First statement says, stratified squamous epithelium is involved in exchange of materials. This is wrong, because stratified squamous epithelium is usually found in places that undergo a lot of tearing and wearing, where a lot of abrasion takes place, usually the anus, vagina, then we have the mouth, the esophagus. So these are places that goes under a lot of abrasion, that lot of work and lot of tearing and stuff. So the stratified squamous is present on those kind of cells and not involved in exchange of materials. So it is involved in. So what about pseudostratified columnar epithelium? So they are saying pseudostratified columnar epithelium is a compound epithelial tissue. So we know pseudostratified columnar epithelium is a simple epithelial tissue. So that's wrong again. Then the third statement, simple columnar epithelium is found in intestinal and nasal passage. Now, we know simple columnar epithelium is involved in secretion, usually involved in secretion and active absorption, right? So, okay. So simple columnar epithelium is found in the intestine, which is correct, but in the nasal passage, usually the epithelium that is found in the nasal passage is the pseudostratified columnar, not the simple columnar epithelium. So third statement is also wrong. In fourth statement, simple cuboidal epithelium is found in the salivary glands and the kidney tubules. Now, if you take salivary glands and the kidney tubules, one of their main function is to secrete substances, right? So simple cuboidal epithelium, so they usually secrete, involved in secretion, and this is correct because simple cuboidal epithelium is found in the salivary glands and the kidney tubules. Finally, we have simple squamous epithelium prevents exchange of substances because, simple squamous epithelium actually allows the exchange of substances. So these simple squamous epithelium are usually found in the alveoli during gas exchange and also in the blood vessels, right? Because gas exchange occurs between blood vessels, right? So simple squamous epithelium are found in these places, and they usually allow exchange of substances. Okay. Now we have seen all the functions of all the cells. So we have only pseudostratified columnar epithelium remaining. So what is the function of pseudostratified columnar? So they usually are found in places such as the airways, nasal passage, because they produce cilia. So the one of their best modification is cilia, and they allow to move the mucus out of the lungs. So fourth one is the correct answer. Now let's go to the 18th question. Okay. 3 types of symbiosis seen among organisms with examples are given below. So we have mutualism, parasitism, and commensalism. So these are 3 types of interactions that organisms undergo. So in mutualism, both the organisms are benefited from each other. Then we have parasitism. One organism is benefited and the other organism is harmed. Then we have commensalism, where one organism is benefited and the other organism is unaffected. So let me write it down. Okay. Now here they are given cow and a crane for mutualistic relationship. So I'll tell what happens between a cow and a crane. You guys decide whether this is a mutualistic relationship or any other kind of relationship. Now, cow and cranes are both found on grasses, right? Crane is a bird. They usually eat insects on grasses. So these cows are also present in grasses. They eat grass. But cranes doesn't eat grass. Now, what does these cows do is, when cow is eating grass, it stirs up the grass, moving here and there. So this causes the insects that are present in the grass to fly up. So when the insects fly up, the cranes notices those insects and cranes go and eat those insects. Now, what kind of relationship is this? So cow has done nothing. So cow is actually unaffected, right? But the crane is benefiting because of the cow's movement. The insects are flying up and the crane is getting its food. So the crane is benefited. So this is a commensalistic relationship, not a mutualistic relationship. Then we have again parasitism, man and planaria. So this is the question, this is the statement we think it is correct, because when we hear the word planaria, we hear the word worms, right? So because planaria belongs to platyhelminths and they are worms. But the thing is, planaria are free living worms and they don't cause harm to the human. So this relationship is not parasitic. So the second statement is also wrong. Third statement, it's about commensalism, whales and barnacle. Now what is this barnacle? So barnacle is an organism that is present in the sea. But the thing with these fellas is that they can't move after they become an adult. So what they do is they go and attach to the whales. When they attach to the whales, when the whale moves, the barnacles also move because barnacles are attached to the whale. And they also get food during that movement. So they don't eat the whale. So if they eat the whale, it becomes parasitic, right? So they feed on other things such as planktons that are found on the sea. So here the whale is unaffected because whale has done nothing. It's not benefited from the barnacle. So it is unaffected, but the barnacles, they are benefited. So this relationship is commensalistic. So the correct answer is the C1. Now let's go through the 19th question. Okay. Select the pairs where an increase of I causes the increase of. This is ready. Okay. Let's go through the 19th question. Select the pairs where an increase of the first event causes an increase in the second event. Okay. First, they are giving stretching of the stomach wall causes the release of gastrin. So let's go from first. What happens during digestion? Okay. Now, food enters the mouth and it enters the stomach. When food enters the stomach wall starts to stretch. When the stomach wall starts to stretch, gastrin is released. So this statement is correct because as the stomach wall starts to stretch, more gastrin is released. Now let's go through the process. Now gastrin is released. What gastrin do? It causes the release of gastrin hormone. So this gastrin hormone causes the release of hydrochloric acid and pepsinogen. So this hydrochloric acid and pepsinogen, what they do? Hydrochloric acid provides an acidic environment for the pepsinogen to work. So pepsinogen converts to pepsin, and this pepsin breaks down the proteins that is present in the food. Now all this happens and it forms something called the chyme. So chyme is the partially digested food. So let's stop at chyme and now let's go through the other statement. Now let's skip the Y statement and let's go through the Z statement. So they are saying amino acid content in chyme increases the release of bicarbonate ions from pancreas. Now, as I said, in the stomach the chyme is produced. Now what happens? This chyme enters the small intestine has small, what do you call it? Not a lot, but a certain amount of chyme is released in the small intestine. Now in the small intestine, what happens is this chyme causes the release of hormones secretin and chymotrypsin. So these hormones, especially secretin, causes the release of bicarbonate from the pancreas. So this statement is also correct, because the amino acid is present in the chyme. This causes the release of causes the secretion of hormone secretin and chymotrypsin. Secretin causes the release of bicarbonate from the pancreas. Now why does this happen? Because on the stomach hydrochloric acid was released, so the chyme is acidic. So the bicarbonate ions can neutralize this acidic chyme. So Z statement is also correct. Now let's go through the Y statement. Fat content in chyme increases the food digestion in stomach. Now we know only proteins are digested in the stomach and the chyme enters the small intestine. Now in the small intestine, if there is a lot of fat present, we know something about fat is that it takes a lot of time to get digested. So in the small intestine, the chyme will be present for a very long time to get digested. Then what the small intestine does is it secretes hormone to signal the stomach and says the stomach, slow down the digestion, fat is still present in the small intestine. So actually the opposite happens here. So fat content in chyme doesn't increase food digestion on stomach, it will actually slow down food digestion in stomach, because chyme is already present in the small intestine and fat in the chyme takes a very long time to digest. So the correct statement is X and Z. Okay. Now let's go through the 20th question. Okay. Select the correct route of blood through the human heart from systemic circulation to pulmonary circulation and back to systemic circulation via aortic valve. We know systemic circulation is the movement of blood throughout the whole body, through the whole organ, or every organ in the body. So that is what systemic circulation is. And pulmonary circulation is the movement of blood from the heart to the lungs to get oxygenated, then from the lungs back to the heart. So that's what pulmonary circulation is. So now let's go through the process. Here I have added an image of the heart. So what happens? Blood goes through systemic circulation throughout the whole body and comes back to the heart. From where does it comes back? It comes back into the right atrium. So let's okay. So this is the right atrium. So they come back into the right atrium, and through the tricuspid valve, this blood is deoxygenated. Now they are coming from systemic circulation. They don't have any oxygen. So what they do, they enter the right atrium through the superior and inferior vena cava. Enter the right atrium, and through the tricuspid valve, they enter the right ventricles. So first the right atrium, then the right ventricle. So from the right ventricle, what they do, they enter through the pulmonary valve into the pulmonary artery. Why does this happen? Because they don't have any oxygen, they have to get oxygenated. Now this deoxygenated blood enter into the pulmonary artery, they go into the lungs and get oxygenated and return through the pulmonary vein into the left atrium. So this is the left atrium. So yes. So from the left atrium again, the blood travels into the left ventricle through the mitral valve. Again, we call this mitral valve also the bicuspid valve. And from that, from the left ventricle again, the blood enters through the aorta using the aortic valve. So from the aorta, blood again travels through the whole body. So first we have now let's go to the answer. First we have the right atrium, then we have the tricuspid valve, then right ventricle, then pulmonary valve, then we have the left atrium, then the bicuspid valve and the left ventricle. So the correct answer is the second one, right? So here we have the right